thanks Ajay for the introduction, and thanks for the whole training team to helping setting up this webinar. So first I'll do an introduction. As he mentioned, I'm Marcelo, and then Joseph will join doing a walkthrough. And today we are going to talk about finding and interpreting protein structure and function data using PDBe KB. And we also have, as I mentioned, a walkthrough of how to access information from KB to answer biological questions. So first of all, I'd like to talk about what is PDBe, what's the protein data bank in Europe. So PDBe is one of the 3 founding members of the Worldwide Protein Data Bank Consortium, which also includes RCSB and PDBJ. Very recently, we've been joined by PDBe India and PDBe China as well. The whole consortium has been going on for more than 50 years. And currently we have more than 200,000 deposited experimental structures. So PDBe mission is to establish a high quality, up-to-date and integrated infrastructure, and also which also includes data analysis tools for 3D macromolecular structures to both basic and applied research, and also for education across the sciences. So you can think of us as the main repository for experimental structural data that allows all the resources and our users to put information into these structures and then to extract actual knowledge to answer the questions they're interested in. So if you haven't taken a look into our pages before, this is what they look like. So here we have a list of all the services that we provide to the scientific community, some statistics on the whole archive, featured article, which is a very nice article about a protein that comes every month. So this is done by our annotation teams and also sometimes by our bioinformatics team. And we also have all the latest news of the whole archive. If you have any molecule of interest, or be it a protein or anything else, you can use its identifier or its text to search on our pages. Then you'll be able to obtain lots of data into them that's from them into the archive. So if PDBe is more about the deposited structures, KB is open and collaborative consortiums that enrich the structural data with functional annotations. So this comes from our various structural biologists and bioinformatics data providers. And right now we have 34 different data resources that contribute annotations to KB from 13 different countries. And all the infrastructure is developed and maintained by the PDBe team. So our annotation providers, we have both functional and biophysical annotations included in KB. So these can be grouped in overall 6 categories. We have biophysical parameters, small molecule sites, protein binding sites, protein end domains, evolutionary conserved sites, and mutations and variations. Some of these annotations, as you can see in this plot here, cover a large fraction of the PDBe archive, while others have a more limited scope, which is completely fine. Sometimes you have annotations that are specific, for example, for phosphorylated proteins, which are very valuable to the community, and we are very happy to take and include in the KB. When we talk about PDBe entries, this might be something new for some of you. So these entries are usually identified by 4-character IDs. We also provide 8-character extension IDs that soon will be the only that we'll provide, since we are reaching the limit of entries that can be identified by only 4 characters. Inside the PDBe entry, we also have summary information about the entry and its molecular consistency. So we have also citation information and details about the experimental determination and the structural validation for this entry. What usually we are interested in is the atomic 3D coordinates for the biological molecular entities that are contained in a PDBe entry. So these are amino acids and their modifications, DNA, RNA, small molecules, lipids, carbohydrates, ions, and water, for example. And with these 3D atomic coordinates, we can visualize the structure. So this figure here on the right shows the visualization of a PDBe entry using a software that our team provides, which is MolStar, in 3 different styles. And also important to mention is that all residue and small molecule components are represented by 3-character IDs in PDB. And soon we also reaching the limit of different residue and small molecule components that can be represented with discrete characters. So we are also including 5-character IDs. So more about what's inside the PDB entry. We obviously have proteins, but we also can have entries that are only composed of DNA or RNA or of all these different molecular entities together, and can be used to study how they interact with each other, for example. We have post-translational modification of residues. So we have an example here of phosphorylation on the left and glycosylation on the right. So all sorts of small molecules. We are talking about small molecules of pharmacological interest, such as drugs, antibiotics, drug-like compounds, also metal ions and water. And you can also have large complexes. So here we have an example of a histone on the left and viral capsid on the right. And these large complexes have been more and more deposited since electron microscopy enables us to solve these very large structures which come to the PDB. So about the format itself, usually what people are more used to is the PDB format, which is our legacy format that has not been modified or extended to support new content since 2012. And its main limitation is that it does not allow to represent large entries. So entries that contain 62 or more chains or more than 100,000 atoms cannot be represented in this format. And when you take a look at the format, what you see is that it's composed of a collection of record types. Each record can contain 1 or more lines and in a maximum of 80 columns. So this figure here is an example of the atom record that people are usually more used to. And you can see the record name in the 6 first columns and the atomic coordinates right here in the middle. So what's our standard since 2014 is the MMC format. It's good because it has no limitation on the number of atoms, residues, or chains that can be included in a single entry. This format is continually updated, so it's extended and improved while ensuring backwards compatibility to previous MMCs and the PDB format as well. And this format can be viewed by a collection of MMCF tokens. So these MMCF tokens are basically underline a category dot an attribute of interest in 2 possible styles. So this figure here on the right shows the key value style, in which you have an MMCF token followed directly by its corresponding value. But what we can also have is a tabular style that allows rows of multiple values. So first you set a list of MMCF tokens, and then you have it's followed by lines which contain data values. There are white space delimited now, so there's no limits on the number of columns. And this makes this format quite scalable and extensible. One example of this scalability is that in our entry pages, so this is one entry page in PDBe, we are including in the download session, you can see here the updated MMCF file. This updated MMCF file includes CFITS mappings. And these are very useful for the scientific community because these 4 MMCF tokens here, as you can see here on the tabular style on the bottom, includes what uniprot protein that residue is mapped to, and what is the residue number on that uniprot protein. So to talk more about KB, which is our main subject today, as I mentioned, our mission is to provide a fair infrastructure, so findable, accessible, interoperable, and reusable data that puts our entries in biological context, and also to develop innovative tools to the scientific community. Our main web service is the aggregated views of proteins, which we'll talk about today, but we also have planned aggregated views for ligands, domains, and complexes. And one of our main projects is 3D beacons, which I'll talk more about in the next slide. But it's also important to mention that our software components are open source to the scientific community, and under the Apache 2.0 license, and that we also provide API endpoints, so both for PDBe and for PDBe KB. And there was also we need to highlight the AlphaFold database, which is sort of a sister project to KB. And we had a very nice webinar last week about that. So yeah, and we also have an FTP server where we provide all the raw data used to build KB. So 3D Beacons is an open collaboration between providers of macromolecular structure models. And this allows unified programmatic access to both experimental and predicted structure models. And these are keyed on UniProtect sessions, so you can get these models. 3D Beacons also support sequence-based search, and data providers include AlphaFold, PDBe, Swiss Model, Model Archive, SAS-BDB, PED, AlphaFuel, and others. So you got the annotations that we provide through KB. Every annotation displayed on our aggregated views pages and our API endpoints, they link back to the original data provider. This is very good because our data providers gain increased visibility and can reach more users. And at the same time, we can provide our users with high quality data and comprehensive data. Sorry. So if you are wondering where this data is on PDBe KB, today we are going to talk about the aggregated views of proteins pages. But if you are more on the programmatic side of things, we also have documentation on our public API endpoints that can be used. If you want to set up the database itself, we provide instructions. So we have a page with instructions on how you would do that. And we also have a schema explorer, so you can see what entities are in our Graph API and how they are linked with each other. If you're interested in the raw data itself, our public FTP access area has data for the annotations, the benchmarking data sets, the superposition data, and more recently, the complex data as well. So now to Joseph.
Okay, so yeah, I'll be walking through some use cases for the PDBe KB. If you'd like to follow along and try this for yourself, there's a QR code in the top right-hand corner here. You can scan with your phone, and then there's also a link in the bottom right-hand corner. So the slides should be made available after the webinar as well, but feel free to follow along as we go. So the landing page for the PDBe KB is what you'll see here if you follow either of those links. And the first thing to do is either search by your PDBe code, so the 4-letter code, or the UniProtect session as well. So when you search this, the first thing you'll land is the entry page here. Now, in this case, I'm using the serine threonine protein kinase as an example. You'll be given here some information on the gene name, the organism, any synonyms for this protein, the UniProtect session, as well as a description of its biological function. And over here on the right-hand side as well, we have a 3D viewer. This is the Molstar viewer that Marcelo spoke about. And then the regions on the corresponding UniProt sequence that are contiguous to the particular PDB chain that you're currently viewing. So, of course, the selling point of the PDBe KB is the extra information that we imbue these structures with. And we do this firstly with a load of structural annotations, which you can find here in the Structures tab, but also some functional and biophysical annotations as well, which you can find on the Annotations tab there. So I'll firstly have a look at the Structures tab. And as you can see here, we have indexed all of the structures corresponding to this given UniProt sequence by residue ID. So we've got residues 1 through to 970, and the first row shows the PDB structures here. So we can find that there are several structures in an N-terminal region and then 2 others in the C-terminal region. We also provide things like structural, secondary structure variation as well, any flexibility predictions, topological annotations, early folding residue predictions, as well as the domains as well, which are given to us by the interpro annotations, as well as CAF. And then in the bottom row here, we've got other structures, and these are the 3D predicted structures from resources like AlphaFold, as well as our other 3D Beacons network partners as well. So if you click on the Annotations tab, this is going to give you more information on the functional and biophysical annotations on a residue-based, a sequence-based view. So again, we index these from residues 1 to 970 along the gray bar along the top. And you've got information such as the curated interface annotations, so which other proteins your query sequence is binding, predicted functional sites, post-translational modification sites, predicted ligand binding sites, any biophysical parameters, again, the domains which are provided by Interpro and Cath from the previous structures tab, and then any actual ligand binding sites that we find in the PDB structure, so the experimentally solved structure, as well as actual interaction interfaces between other proteins in the complex, as well as sequence conservation and variants as well. Now, you can also access the PDBe KB from the PDBe website as well. So in the top right-hand corner here, you can see the search bar. And this is where you can search for the PDB code, the name of your protein, any ligands it might bind, the gene name, the uniprot, and more. And if you would like to specify and provide a more advanced search, we have a button for that as well, just below the search bar. So when you first make your search, you'll find a list view here. Now, I've only shown the first hits, but in this case, you can see there are a total of 344 hits for this search. And I'm just showing you an example here of a crystal structure for CAMP-dependent protein kinase. And the first tile will give you some information here on things like the authors, the source organism, any bound ligands, modified residues, etc. And importantly, along the bottom here, if you would like to now go and navigate to the PDBe KB, then you can do that by clicking on either of these two links. And in this case, there are two proteins within the solved structure, and these two links correspond to those proteins individually. Now, if you would like to search for the complex, so other deposited structures which have the same or similar subunit composition, you can also search for this now unique complex ID too. If you would simply like to find the best structure, then click on the macromolecules tab, and this will show you, again, a list view of the best structures for your search. In this case, the top hit from before is the same, but again, you can navigate straight to the PDBe KB page using either of those two links here on the bottom left. So I'm now going to walk through a few use cases. This is by no means an exhaustive list, but it simply gives you an idea, an appreciation for some of the questions you might be able to answer using the KB. So the first one is, what ligands does my protein bind, or what ligands bind my protein? And again, as I said, you can access this either from the PDBe website directly, or go straight to the knowledge base, make your search, and you'll enter the entry page over here. Now, if you make your way over to the ligands tab along the top there, you'll find this tile-based view where you can find all of the ligands which have been found to bind your query protein across the PDB archive. And any cofactor-like ligands we label here in green to help your search. Now, if you scroll down from the page, you'll also see a uniprot sequence viewer as well, where you can see the ligand binding sites for all of the ligands within the PDB archive. Now, if you click on the ligand binding sites, you obviously get a lot of hits here. However, just to point out, these two bands here, these two bands in the heat map, correspond to the cofactor-like ligands. So you can use this to really interrogate all of the structures from the archive for this given protein in one place. Okay, so another question might be something like, what complexes does my protein form? And again, you would probably navigate to this either directly from the PDBe website, or you go to the PDBe KB page, click on either of these two links. That would take you to the entry page, and then click on the Interactions tab here. Now, the Interactions tab shows you all of the other proteins with which your query protein has been found to be in complex with. And you can click on any of these links, and that will take you to the corresponding entry pages, so the summary page for these given proteins here. And then if you scroll down, again, we give you a sequence-based viewer for this particular search, where the table shows you all of the interactions for your query sequence, your query protein, against all of the other proteins which are found to be in complex with it across the archive. So thirdly, another question which you can now answer on the PDBe KB is, what conformations does my protein adopt? So if you navigate over to the summary tab and click on the button in the bottom left here, you'll find the 3D view of superposed structures tab, and if you click on that, that will open up a MolStar viewer. And just to clarify what some of these things mean here, if you click on the Select Segments button, this will open up Select Segments button in the left here. This will open up all of the rows that correspond to the potentially discontiguous regions across your UniProt sequence with which we have PDB structures. On top of that, we also cluster the sequences based on their 3D coordinates. And these are essentially a proxy to our recognized conformational states based solely on 3D coordinates. And as you can see here, for the first segment, we find 2 clusters. So to give you some more details here on what the 3D viewer is showing you, this is a MolStar window. I'm now looking at the third segment here because this has a really nice conformational change between the orange subunit and the green subunit. By default, we only show the representative chains, which are the best modeled structures from the archive. But now you can also load in the AlphaFold structure, which will be imported and superposed on the fly. So once you click on the Load AlphaFold Structure button, what you'll have here is the AlphaFold structure superposed to the corresponding experimentally determined structures as well. I'd just like to bring your attention here to the bottom right-hand corner on the right-hand tab, where we have the AlphaFold's superposition. And you can see here the RMSD of the AlphaFold structure to 5LHZ chain A is very close in comparison to 5LHY chain L. Therefore, it sort of corresponds to the fact that the AlphaFold structure is much more similar to the structures in cluster 2 than it is to the structures in cluster 1. We also show PL-DDT here in sort of a high PL-DDT in dark blue through to low PL-DDT in orange, to give you an example of sort of the quality model prediction, which also acts quite nicely as a proxy for integral disorder, actually. We also show the PAE here on the right-hand side. This is the predicted aligned error for the AlphaFold prediction as well. And then lastly, does ligand binding affect protein conformation? This is more of a compound question, but if you navigate to the Summary tab and then click on the 3D view of superposed ligands button in the bottom, this will also open up a similar MolStar viewer. This time, though, we're trying to emphasize and trying to show where the ligands are binding in these clusters. So you might find that some ligands are binding more in cluster 1 than they are in cluster 2, or even exclusively in one of these clusters. And you can also load in the AlphaFold structure as well. So we only show the representatives by default, but you can at least see where all the ligands are binding. So if you would prefer to access this information programmatically in JSON format, you can also do that using our Graph API. We're currently migrating that to a more of an aggregated API, but this is more of a reference just to show you that you can access the same information programmatically as well. And finally, if you would like to get all of the annotations that we have for a given PDB ID, so a 4-letter PDB ID, you can use this URI here, this API endpoint, in order to get all that information, again, in JSON format. So just to recap everything, really, the PDBe, so yeah, us at the PDBe KB, we aim to provide a fair infrastructure so that you can access all of the information that we have, both via your browser and programmatically as well. We want to provide you, the users, with relevant structure information, both at a sequence level and a 3D level too. Hopefully you'll find that you can quickly navigate to the information you need at a glance, and maybe then download it programmatically if you need to later. And we also want to give you access to those detailed structure annotations via our graph database, or accessing it through the API endpoints, like I said earlier. So if you would like to, you can download the entire graph API, the entire graph database, and run Cypher queries on it, because it's a Neo4j database. So for your reference, these are all the links to the different resources that I've been talking about. Our FTP area tends to contain lots of raw information, especially for cluster annotations and things, as well as some benchmark data sets that we've curated for different purposes. So we currently have 2 surveys running at the moment. If you'd like to help us design our new macro molecule complex pages, either use the QR code here, or you can follow the link when the slides are made available. And then on the right-hand side, we've also got a ligand-related API that we're working on. So if you'd like to share your thoughts and contribute, then please have a look at those. And if anyone has any questions.
All right. So to start with the question, I think, Joseph, you already started typing in answer for one of the questions.
Yeah, so I think it's is it Marcus Lopez?
Yes.
So Marcus Lopez asks, are there different entries for PDB and PDBe? So the short answer is, I'll stop sharing my screen actually. The short answer is no. The PDB files themselves are the same across any of the databases that you use. So whether it's us at PDB or RCSB, PDB or PDBJ, the PDB files themselves are the same, and that goes for the PDB codes. Now we have a few other data formats. For example, the updated MMSIF file contains uniprot mapped sequence IDs. So this is particularly useful when you've got different numbering schemes between PDB files or MMSIF files from different groups. And if you map them all to their corresponding uniprots sequence, then it sort of standardizes the sequence then. So that's one thing we provide, but I think you can access it from the other databases as well.
All right. And there's another question I think you already answered in your presentation probably. Please, I want to know how to retrieve protein and nucleotide sequences. I need step-by-step guide. So I think it was part of your...
Yeah, so when it comes to the protein sequence, you can access that from the PDBe website as well. I think we might have graph API endpoints for that too. But if you head over to the PDBe, we have it in faster formats if you'd like to download the sequence.
It's a PDBe endpoint. We can get the sequence.
And you can get it from the yeah.
Okay, great. Does the database need multiple structures for the is it conformational or conformations clusters to be useful? Can we still use it with just one structure resolved in each conformational state?
Yeah, that's a really good question. So at minimum, you need two structures to really determine the conformation. We try to, if the structures are sufficiently similar, then we will group them together as a single cluster. But if there are any differences between them, we'll try to separate them. But the more structures you have to cluster, typically, the better the recognition of, I guess, what we would say maybe a true conformational state is. But this is something we're kind of working on more actively at the moment. So yeah, the more the better.
Great, thank you. What is the early folding prediction?
Well, I can answer. So the early folding prediction will be something that one of our partner collaborators provides us with. So if you go to the KB website, you can find links to all of those, all of our, I think it's 34 collaborators now. So that's something that they provide us with. I'm not expert on this, I'll be honest. So I can follow up if you'd like a more detailed answer on that. But the short answer is you can find our collaborators page on the KB website.
Great. Why might there be a discrepancy between structures in PDBe and AlphaFold? Or are there discrepancies?
I don't mind. Marcelo, do you want to take it? Oh, I'm happy.
I think this is related to superposition, actually.
Yeah, I mean, it's a great question. The short answer is the AlphaFold structure is a prediction. So it has its evoformer model. Yeah, it's an interesting question. Sometimes they're different. I mean, in the PDB, we often find that when some structures are ligand bound or in a different complex, or maybe there's a single point mutation, then that can have quite a profound difference in its structure. Sometimes it's not, it's quite context dependent. But I think just being able to see the differences is quite informative. So yeah, AlphaFold gives you a single static structure. And I guess to add to that as well, in the AlphaFold database, we have, I think it's 213 million structures. So it's basically the entire uniprot. No, sorry, there's a lot of sequences. So a lot of sequences for which we have structures for. Now, we only show the top hits. But of course, if you run AlphaFold, you get a range of models as well. if we were to show maybe the top 5 or have the top 5 in the database, maybe you would find the 4th hit, for example, gives you a better superposition with one of the other conformers.
Yeah. Another thing to take into account is that the AlphaFold entry will contain the structure for the whole protein, the predicted structure for the whole protein. So there might be some detail you'll be interested in when you're doing superposition. And
yes, this is very true. Yes, so often for lots of different reasons, there might only be a single region that has been solved. So maybe it's just a single domain has been resolved, and that will fold up in one way. But if you consider the rest of the sequence, that might have an impact on it too.
Thank you. Do you have a tool to compare protein structures with different sequences and highlight the differences?
I think we don't have anything at the moment, but this is something that's kind of planned for the domains pages and maybe the complexes one as well. What do you think, Joseph?
Yeah, so it would be nice to maybe go down to something like 90% sequence identity. And that would obviously expand your structure space. But we don't have a tool for that yet. So right now the KB pages are indexed by UniProt ID, unfortunately. But yeah, this is something which would be nice. Yeah.
Okay, great. Is there something where you can superimpose multiple structures and then see where they are not aligned properly?
Yeah, so I didn't show this on, if you, on our pages currently, we do that for sequences which have 100% sequence identity. So on the left-hand side of the Molstar viewer, you can reveal all of the structures and they'll get loaded into your browser instance. Of course, if you've got 100 structures, that can slow your machine down a bit. So I only showed the top hits, but yeah, you can show all of them and find any which might have a discrepancy or similarities.
Okay, great. How to get to know the tunnel analysis for any protein in PDBe KB?
So I'm guessing this is related to the annotation tabs in KB as well. So if you go to the annotation tabs, there will probably be the annotation for the tunnel analysis. And if you want to know more about it, then you can access the data provider and see how they did it, actually.
Yes.
But we'll be happy as well to help you if you want to send this question. We can go through the details as well.
Yeah.
Yeah. So
I would say on the structure annotations tab and then the biophysical or functional annotations tab, if you click on the rows, that will give you a drop down of the more specific annotations which are given there. But to fit it all on a slide, I had to collapse them.
Yeah, that's great. Yeah, and just to add to that, if you have a very specific question about your protein, you can send it to help desk PDBe.
Absolutely.
Okay, great. The ligand which interacts with protein are made available based on already available data in literature, or it does so other than what's in the research papers. So do you get something, I think, essentially, which is not published in papers as well?
Okay. That's a good question. As far as I'm aware, no. So the ligands will be from the corresponding PDB structure. The extension to that are predicted ligand binding sites, which are provided by our partner organization, our partner collaborators. But they come from the PDB file themselves, or the MMSIF file.
Okay. Thank you. Can we dock 2 proteins with a single ligand at one time?
It depends. It depends whether this person means dock as in predicting whether the ligand will fit into a particular binding site, or whether it's already been experimentally determined in a particular binding site. So to answer the second part, yes. If there's a ligand present in the structure file, then it will be there in the ligands button. But docking the single ligand into a predicted binding site would be more of a research problem. So I don't think we have the data for that.
Right. Thank you. If proteins have missing information, then is it possible to fill it and predict structure of that protein?
I think that one is related to the one below that says new identified ligands are also there. And it would, you can check the 3D Beacon's website and also the AlphaField project which for predicted structures, we try to fill up with new information such as ligands, for example.
Yeah. And I guess to answer that first question there as well about filling in missing regions, if it is in terms of model coordinates of the peptide, that's why we have the predicted models along the bottom row as well. So of course you've got the AlphaFold structure too, but we also have other predicted models too you can use to fill in the blanks if they have been modeled in the experimental structure.
All right, next question is, could you explain how to overcome the loops in protein structures?
I guess I can speak from a clustering perspective. So the loops might be informative if there are regions which are in a different position. That might be something we want to capture and highlight. But maybe if there's more of a specific use case this person's referring to, I can answer that.
Okay, great. The next question I think we have already answered is about step-by-step explanation about retrieving protein nucleotide sequence. The next question is there any tool to phylogenetically analyze the sequences or alignment?
There are such tools, but this is not really related to what we've been doing at KB, not at the moment. It might be something relevant, especially in the domains ones. But yeah. And
if you want to organize it by sequence, if you want to arrange your sequences by phylogeny, you can run something like a multiple sequence alignment as well. It becomes more difficult when you're considering on a structure basis. But yeah, if you want to run multiple sequence, there are lots of different multiple sequence alignment tools, and you can use this for exactly. Yes, someone's just, but yes, Cluster Omega is a good example. Yeah.
Yeah, and to add to that, we have webinars from Uniprot and Interpro.
Yeah, they could probably speak about that more
in coming weeks. So please join that webinar, and you will get more details about that.
Yeah, and for the first question about the step-to-step explanation, we also have nice tutorials in the front KB pages that can be used for this sort of purposes. So
yeah, especially if you want to access a single file. Yeah.
Yeah. Okay, great. Is there anything specific related to protein misfolding and stability study in PDBe?
In KB, we have annotations for those from one of our partners. So if you go to the annotations tab, you'll probably
yeah, the AlphaFold structures as well can, if you have low PLDDT, that can be a good proxy for disorder as well, the structural disorder. That might be important for misfolding because that will often be how they begin, and then they'll become more stable when they're. I don't know whether they're referring to like prions or aggregates in this case.
Yeah, for our partners, there is Dynamine, which predicts disorder, especially. So I think they also have some parameters for misfolding. I'll have to double check, but it's one of our partner resources.
Okay, is it like a tool or is a resource?
These resources are part of KB, so they are mapped into our structures. So we provide API endpoints, and also in the aggregated views of protein pages, you can see in the annotations tab more about that.
Okay, so you don't need to specifically go to Dynamite pages and to look for it. Okay,
you don't have to map it yourself. It's already mapped in KB.
All right, that's good to know. What's the current version of AlphaFold PDBe uses?
Good question. I'm not sure what the current. I
think it is the latest one, definitely.
It'll be the latest one. Yeah.